And let's start with Dr. Nazan Gunders from Philip Morris, who's going to come and talk to us, and much looking forward to it. Thank you.
Good afternoon. I'm a bit loud, maybe. Before I start, my name is Nazan Gündüz. I am leading oral smokeless category in PMI from Life Sciences. I would like to answer the Japan question first. I was not going to present, but I was able to get the answer. And this is in our earnings, Q1 earnings, that we reported for Japan, that overall heat-not-burn category, so these are smokeless categories. I don't have the total of the smoking, but the 35% of the total tobacco is with the heat-not-burn category. So of that, IQOS is leading the category of that majority of the 35%. I have the Tokyo numbers, about 32%. And the different cities. So this is definitely linked to the strong performance in Japan, linked to the innovation, technology, and also understanding the consumer needs, right, as we mentioned, which is very much the topic of my talk today. So with that, I'm going to switch to my talk. So I'm very excited to be here. This is my first GFN talk, so hopefully all will go well. So the talk of my conversation today is about innovation of reduced risk alternatives, specifically focusing on the oral tobacco products and nicotine pouches. So in PMI, our main goal is to really to build the smoke-free future. This comes with a portfolio of products where the scientific substantiation is achieved and with a portfolio of options that we provide to the consumers. It is very important that we do come with a robust product design that starts with the development, understanding all incoming ingredients, understanding their toxicological the attributes, and to be able to design portfolio of these options, the product options. We place the controls in terms of the chemistry, in terms of the manufacturing, in terms of the supply chain, to make sure that unintended events are eliminated, otherwise minimized. Only then, with the scientific substantiation, with the robust science, then we can talk about the harm reduction, exposure reduction, harm of risk, and disease reduction. And we can also have dialogue with regulators. So it is very much in the center of everything, that robust product science. And if we are able to combine the science and scientific approach to the product attributes and the consumer acceptance, then we have an opportunity to talk about the harm reduction. And so harm reduction, of course, it comes, as I mentioned, that consumer acceptance. We need to be providing a portfolio of products to our consumers so they have options to choose from one or many of these products from the portfolio. And we need to make sure that the substantiation is there to be able to report on the risk of harm and risk of disease reduction. So with that, if we have more of these smokers accepting the reduced risk products and switching to those products, then we can look into the totality of the population harm reduction as a whole. So there is if we are able to combine the robust product science and then the assessment strategies, then one can achieve the reduced formation, reduced exposure, risk of harm and risk of diseases, starting from the chemistry all the way to the post-market studies and epidemiological studies. And with that, in PMI, we do have a standard approach for all of our smoke-free products. So although I'm going to be talking about the oral smokeless category, which is a risk-based approach that we apply, but it is very much at the design of the quality by design principles. When we look into the overall the product and then the control parameters, looking into the toxicological assessment of the ingredients, both for the formulation at the formulation level as well in the finished product level. Looking into preclinical assessment, right? Looking into the in vitro studies, cytotoxicity, genotoxicity of these products. Clinical studies also give us the great data to see how well that these products are, how products are consumed and how well that they are actually satisfying the consumers, both from the pharmacokinetics and pharmacodynamics point of view. And finally, the consumer perception studies and the post-launch real-world evidences can also tell us about the harm reduction potential of our products. I have a bit difficulty with the clicker. Okay, so not all products are the same, right? I think we appreciate that. Not all tobacco and the nicotine products are the same. And when we look into this risk cliff, you can see that there's a substantial difference, this is for illustration purpose, between the combustible products and the non-combustible products. The significant difference, of course, when we talk about the non-combustible products, that, and especially for the pouches, nicotine pouches, the absence of tobacco, the absence of combustion, heating, and so that, and also the ingredients that we use, the food-grade ingredients and the pharma-spec-grade ingredients, one can clearly differentiate the differences in risk between cigarettes and these non-combustible the products. And when we go into the details of this now, nicotine products, so overall, oral smokeless categories, they are not new, right? When we think about especially snus, the loose tobacco, snuff, they are not new. They have been around for centuries. But the nicotine pouches, they are new, innovative products in the last few years, especially in the US. You will hear from my colleague from Brian, the US, the data as well, has been successfully accepted by the consumers. So the significant differences between nicotine pouches and the traditional snus, or in general snus, I would say, the absence of tobacco, of course. And you could also see from the visually that lack of tobacco, no tobacco, also gives the better hygienic options for the consumers. In terms of the harm reduction potential, you would already maybe aware of that. The Swedish match snus in the US is achieved the first MRTP approval from FDA. So this is a great, I think the data, the great product compared to cigarette, of course, that both from the supply chain for the quality of the ingredients and the practices at the agrochemicals, I would say tobacco level, and then the practices at the manufacturing, the control qualities that gives the general snus that the MRTP approval. You will hear more on this from Brian as well, but this gives us the great data to compare nicotine pouches to snus, to general snus, as well to cigarette smoke parameters. So, about nicotine pouches. If people are confused, I think, about the nicotine pouches versus the snus, we can easily say that the nicotine pouches, they have simple formulations. They have simple compositions. When you look into the nicotine pouches, I hope you did get a chance to look into that outside in the booths, that they include cellulosic material with nicotine. And they are often, in PMI, we have a tobacco extract nicotine that we use, is a pharma spec grade. It includes the fillers, the binders, polymeric resins, The pH and sweeteners. So these are like a flavoring compounds. All comes together in a pouch material, in cellulosic pouch material. So that's it. So technically, what you get in, what you see inside, and what you get out. So in terms of the chemistry and then the toxicological analysis, we make sure that we follow our internal standards as well externally established voluntary industry standards available for both nicotine pouches, comes from for instance, Swedish Institute of Standards, as well for snus, GOTYETEX standards. These are established, especially for the GOTYETEX standards, established already some years ago by Swedish Monopoly, and then with the Swedish Match, gives us the great reference to compare nicotine pouches against snus. Again, as I mentioned, the cigarette smoking as well. So both of those standards, they give the guidance regarding to supply chain requirements, regarding the manufacturing quality requirements, for GOTYETEX standards for agrochemicals. Incoming tobacco qualities are super important, and as well for the labeling. So we ensure that the labeling, the packaging also follows the CLP guidelines, depending on the regulations, and then we follow the limits in terms of the pH, in terms of the nicotine levels. As I mentioned, all the ingredients are the food grade, and then the nicotine is the pharma spec grade. So it is a simple formulation that nicotine pouches specifically entitles to. So when we look into the toxicants, right, it is important to understand that when we compare nicotine pouches specific to the cigarette, that how significant the toxicants are reduced in nicotine pouches. So the study that we have done, I know some of our colleagues in the room that they have also done the similar studies, that what we did, we compare our nicotine pouches against cigarette, the reference cigarette, as well the general snus. So we did follow the WHO 9 toxicants and look into the chemistry. So all of the toxicants, they were either below the quantifiable or detectable limits. And as a result, we have concluded that nicotine pouches, they have more than 99% reduced toxicants with respect to the cigarettes, and far lower levels with respect to the general snus. We have also done tooth discoloration studies. These products, oral products, right? They are placed between gum and the cheek. And it is important that for the consumers that it also meeting the hygiene, the properties, the requirements that they have. So we looked into the tooth discoloration of attributes of these products, and we used actual human teeth for this study, and exposing the teeth for about 16 days, brushing daily to the extract of the nicotine pouches without diluting. So we have looked into what happens before and after 16 days, and we used the snus, coffee, as well as cigarette for the reference products. So all of our products that we concluded that the nicotine pouches, PMI nicotine pouches, do not cause this discoloration, the tooth discoloration, which is an important for our consumers of nicotine pouches. From the clinical studies point of view, I mentioned that the nicotine uptake in bloodstream, it is very, very important for us to be able to understand that how the nicotine delivery performance of these products, and also potentially the craving, right? So whether or not we are meeting the craving reduction in our consumers' use of these products. So what we have done here, so we looked at 6 products. Some are commercial products, some are prototypes, and we have run the 30 minutes the clinical study using our products. So we used 24 subjects for these studies, and it's not by design, but there happens to be none of these consumers, in this case, tobacco smokers, but not the pouch users. So we have seen that both the speed of nicotine delivery and overall the concentration of the nicotine, it was less than the tobacco, the cigarette actually, the cigarette PK, which we have gathered from the literature. So our objective here was to understand that what is the impact of the nicotine, what is the impact of the pH, the moisture, as well the pouch format. So in our kind of a design of experiment, we have these variables that we want to understand, not only from the nicotine delivery point of view, but also from the perspective of the pharmacodynamics. So what you see here is that all of them, they really follow the similar curve. From the nicotine delivery point of view, and Cmax did not exceed the cigarette smoking. So this was also what we were expecting. In terms of the Tmax, the maximum time was linked to the duration of the product use, which was around 30 minutes. So all of the subjects, that 24 subjects, they did report that the craving reduced after product use. So they did not look to smoke, basically. And then we didn't see any safety signals, which was also great, and we were not expecting anyway, we were expecting no safety signals. One of the things that I wanted to highlight, that not all nicotine was extracted from the pouches. So again, if you have seen some of the publications, which is quite interesting, that when our consumers, they use these products, they use 30 minutes, they can use a bit longer, they can use less, like 15, 20 minutes, but good 40 to 70% of the nicotine, it remains within the pouch. So not all the nicotine is extracted after the use. So this also important to highlight, and this linked to as well that the curve that we see. So we proved that as well. So we took the samples after the clinical study, and we extracted remainder of the nicotine from the pouches, and this was our results, that about 40 to 70% of the nicotine remained in the pouch. So, and finally, from the pharmacodynamics point of view, we have looked into the liking and acceptance of these products by the consumers, the subjects that we have. So it was interesting, I would say, that the consumers, I mean, what is interesting is that it doesn't mean that you have a higher nicotine and the consumer is going to be liking the higher nicotine product, right? It does not work like that. Not only that the products are different, right, and they perform differently, our consumers are different as well, right? They react differently to different product. So we have seen that the consumer respond differently to nicotine strength. And sometimes the lowest nicotine product option was liked the most, but depends on the moisture level and depends on as well the pouch size. It was critical. So what we have seen in the PK curves, the highest like Tmax, I mean the Cmax, did not correlate to the pharmacodynamics. So that also highlights that we need to design products and we need to provide a portfolio of options to our consumers to be able to make sure that we meet their needs. Regardless of the strength level of the nicotine, all of our products address the craving. So they did see the reduced craving and they delivered the satisfaction. And when we look into this exactly subjective effects, which are self-reported, that we were able to judge that how we can design our products from the perspective of the pouch size, from the perspective of the moisture, as well by varying the nicotine levels. So we have reported our clinical study in the clinicaltrial.gov, so you can definitely feel free to look into that. So with that, I will be concluding my summary here is that it is our intention in PMI to definitely to drive the smoke-free future. And that only comes by providing the alternative of products and scientifically substantiated reduced-risk products. And that, of course, we have to make sure that the robust science, and we do have the robust assessment approaches to be able to link and have a claims associated with that. We realize that, I think all of us realize that not all tobacco products and all nicotine products are the same, right? We need to make sure that we do have the low-risk options, and we also design to provide these options to the consumers so that we enable switching to those less-risk options. And our assessment approaches today are designed to basically, for all smoke-free products that we have in our portfolio, that follows the same principle by design. So with that, I am ready to take some questions.
Thank you very much, Nazan. Thank you very much indeed. That's very good, very good. I can't see a thing here. Julian, I can see you.
Nazan, merci beaucoup for a great presentation. Congratulations. Question on your Tmax. So you say the Tmax is 30 minutes, right? Mm-hmm. In your trial, how did you educate the patients for them to understand that it took some time for the nicotine to kick in, right? Because it's very different from a conventional cigarette where you have something kicking in within a few minutes. Question number one. Question number two, and maybe you're not going to answer, but is there going to be any sort of development undertaken to make sure that the Tmax becomes faster? Because that's basically the key to making it work and go broad beyond the Nordics or the US. Merci beaucoup.
Merci. So first question, so we did have some, of course, briefing with our subjects prior to the trial. So explain them about the product and explain them how they should be using. For instance, we didn't ask them to move the product around their mouth. We asked them to park the product for 30 minutes. And we did follow the scientific publications as well when we decided the 30 minutes or maybe 60 minutes that in some of the publications that you might see. So there was, of course, briefing, explanation, and what they expect to see. But without influencing, of course, too much, because that would be not so good outcome for the pharmacodynamics that we were looking for. We, again, as I mentioned, that we always look for the tobacco users when we do the clinical studies. We do have a protocol. And so not by design, but by chance, all of our consumers in that study, they were non-nicotine pouch users. So it was new to them as well. So I am sure a bit like they had to experience a little bit during the clinical study, but with the briefing and then with their feedback that afterwards, and then maybe the questions as well after, this is how we manage. Regarding the second question, the Tmax, I mean, this is an interesting question. And there are also, as I mentioned, that the product designs, and then we are looking into like what is the ideal product perhaps, but there is no one ideal product, right? There are ideal products for our ideal consumers. And so, but sometimes it's like, reaching the Tmax, like, I don't know, too soon may not be the best option, right? We need to see. But definitely, I think by looking at the different product options, that one can play potentially advancing the Tmax earlier. But again, if you were to keep the product 60 minutes, then your Tmax is going to be 60, right? And then if you go to 15 minutes, your Tmax is going to be 15 minutes. So.
Thank you. Jonathan.
Thanks. Yeah, John Fell from the UK. I think you mentioned that your product uses tobacco-derived nicotine.
Correct.
Does it make any difference from your perspective if the product uses tobacco-derived nicotine versus synthetic? How do you think about that?
Yeah, thank you very much. So in PMI, in all of our products, nicotine pouches, the 100% tobacco extract nicotine, and we do reach the pharma spec grade, and that's definitely the requirements that we have. So we have not looked into the synthetic nicotine, and we have not looked into from the nicotine PK clinical trials point of view. So I would expect that in terms of the performance, perhaps that similarities. I cannot judge. Maybe I will ask Brian or my colleagues here. They have maybe a bit more longer experience. But yeah, I think PMI's policy to go with the tobacco extract, and as long as we meet fully the quality requirements, and we do.
I'd like to score well on whatever speaker surveys they do at the end. So if you don't mind, I'll bring that to an end. Otherwise, I'll run over time and I won't score well. But that was very good. Thank you very much, Nazan. And
thank you very much.